Afternoon, grade 10s. I'm Mrs. Van Sale, and I will be doing with you Life Science grade 10 work on the cell structure and the functions overall on the cell. So if we're going to start, you will see that we're going to do this in a part 1 and a part 2. The first one, just talking a little bit about the cell. The cell theory was developed in 1839 by microbiologists Sleeden and Shawn. And I think it's very creative and how they had to discover it with all the little technology that they had and the microscopes that they had to develop, etc. So if we're going to move over to our slides, we will see that the cell theory, as I said, was discovered by Sleeden and Shawn. All living things are made up of cells. All living things have a cell, and cells are the basic unit of structures and functions in the organism. Basically, it makes up the basic unit of life. Cells comes from the reproduction of existing cells, cell division, and you're going to later do that in more detail when you do mitosis. The definition of a cell is the smallest part that is capable of performing life functions. And how incredible is that not? For a cell to be able to perform all life functions. Now here's just a few examples of cells. You will see the amoeba, a type of plant cell, a bacteria cell, a nerve cell, and a red blood cell. All of these are extremely small, microscopic small. And for you to be able to see it, you have to use a scanning electric microscope. Then if we're going to move on, we get two types of cells, a prokaryotic and a eukaryotic. The prokaryotic has no nucleus, where the eukaryotic contains a nucleus. That is one of the primary differences between a prokaryotic and eukaryotic. If we're going to look at a prokaryotic, they do not have structures surrounded by a membrane, not at all. They have few internal structures, you will see comparing to the eukaryotic later, and one-celled organisms such as bacteria, etc. You're only going to discuss bacteria in much greater detail in grade 11. So for now, we're going to basically focus on eukaryotic cells. Now, eukaryotic cells contain organelles surrounded by membranes. Most living organisms have eukaryotic cells. And you will see that there's only two different ones, which is the plants and the animals. Cells that have a nucleus and membrane-bound organelles are eukaryotic cells. They're more complex, not single-celled or one-celled like the prokaryote, and you will see that they have lots of different parts that takes place. So the eukaryotic cell contains three basic cell structures. The three basic cell structures is the nucleus, which is extremely important, especially in grade 12, cell membrane, and the cytoplasm with organelles. Now to start, this is just an idea of what an animal cell looks like. You will see that it's roundish, it has a nucleus, ribosomes, mitochondria, and it has a basic shape as well as a typical plant cell. But you will see that the plant cell have a few things extra. So we're going to go into detail into all of these things and their functions and what part they play in the cell and for us to do our everyday work and life things. So organelles are very small. As I said, you can only see them with the scanning electron microscope. So please guys, you cannot see this by eye. Don't even try. They can perform various functions for the cell, and they are found in the cytoplasm. May or may not be membrane bound. Now if you're going to look at that, the outer membrane of the cell that controls the movement in and out the cell is doubled layer. Now the basic thing that we're going to start from the outside in is the cell wall. The cell wall is only in plant cells than bacteria. The outer membrane that supports and protects the cell, it is made up of cellulose. It's a non-living layer, and it supports and protects the cell. The functions of the cell wall is it exchanges, and all the exchanges between the cell and its environment have to pass through this membrane. It controls, helps with osmosis and all of that. Then the protoplasm refers to the living material. Protoplasm is made up of cytoplasm and the nucleus and all the inclusions on the inside. Osmosis is the movement of water in and out from a region of higher water potential to a region of lower water potential across a semi-permeable membrane that separates the two regions. Now, this is vital for plants and animals' survival. Just because in plants, osmosis helps to absorb water from the soil and transports it to the leaves. Where in animals, for example, kidneys, keeps the water and salt levels in the body and blood at the correct values. So it's not too high or too low. It keeps it at that specific part that needs to be controlled in the body or in the plants. Now the nucleus is quite an exceptional part in the cell. It's the largest organelle in the cell. It distinguishes between an animal and plant cell. You're going to still see. The function is it controls gene expression and facilitates in replication. Now by doing that is what you're going to do in grade 12 much more is it contains the genetic material, the DNA of us, and that is how we can distinguish from parents and offspring and how we know and how it makes you original. A structure also contains the nucleolus, and it's found inside the nucleus. Just a little bit more on the nucleus. Each cell has fixed number of chromosomes that carry a gene. Like in humans, we have 23 chromosomes, and if we have one extra or one less, it's called a gene mutation. So every organism has a certain amount of chromosomes, and that makes you unique to yourself. Gene controls cell characteristics. Also, the DNA contains your genes, and the genes contains your eye color, your type of hair, your color of your hair, your length, your characteristics. So it contains a whole lot of extra things that does not necessarily you can see on the outside, but also your personality, etc. This is just an idea of what it looks inside of the nucleus. You have the chromatin network, but if you unwind it, it forms the chromosomes, which are extremely long. And all of those chromosomes is your genetic material found in the DNA, and that makes up your body and you as a person. DNA is spread out and appears as chromatin. As we said, it's non-dividing cells. Please, guys. DNA is also condensed and wrapped around proteins forming as chromosomes in dividing cells. So it's lots of things wrapped out. So if you unwind it, it's going to be long, meters long even. Then the nuclear membrane, also called the nuclear envelope, surrounds the nucleus, and it is made up of 2 layers. It also has openings which allows material to enter and leave the nucleus when it sees fit. Chromosomes, we just spoke about that as well in the nucleus. It makes up your DNA, it makes up your uniqueness, your character, the way you maintain yourself, etc. And it has the instructions in your body to tell them what to do when and how to grow, etc. The nucleolus is inside the nucleus. It contains RNA to build protein, which you need to build amino acids. It disappears when cells divide, and it makes ribosome that makes proteins. You will see that it has pores, it has an envelope on the outside, the nucleus, the nucleolus inside the nucleus, and those are your chromatin network around there, which unwinds for your DNA. So it's really quite magnificent. And please remember again, this is microscopic small. So if you see it like this, it's amazing to think that each cell contains a nucleus like this with certain characteristics, and your whole body is full of that. Then the cytoplasm. The cytoplasm is a gel-like mixture. It's all inside the cell that all carries all the mitochondria, the vacuole, the nucleus, etc. It's surrounded by cell membrane, which keeps it intact, and it contains the hereditary material, which means that we can see our trace our father and our mothers and our gene line. It also provides a medium for chemical reaction to take place. So it's your liquid where the chemical reaction will take place from one part to another. Then the endoplasmic reticulum, that moves material around in a cell. You will see that you get smooth endoplasmic reticulum and rough. The smooth synthesis of lipids, oils, steroids, etc., where the rough do protein synthesis and membrane production, which is really quite interesting. The smooth also, you'll see that it's smooth on the outside, lacks ribosomes, where the rough has ribosomes embedded on the surface. So you will see the round structure on the outside of the endoplasmic reticulum, and that is where it's rough, not as smooth as on that side. Ribosomes, as you just saw in the previous picture, the rough part for the endoplasmic reticulum, each cell contains thousands and thousands of ribosomes. Ribosomes also plays a role when we do protein synthesis and by making proteins. It's found on the rough endoplasmic reticulum, and it's also found on floating throughout the cell. Wherever you will see it, you will see some of the ribosomes inside the cell. Then the mitochondria. The mitochondria, as I call it, is your powerhouse. It gives you the energy to do all the different parts of the cell. It helps you with transferring nutrients. It also helps you to store large amounts of proteins, which helps us with cellular respiration. It helps us with energy production, which is for ATPs through chemical reactions, breaking down fats and carbohydrates, and for you to be active. It also controls levels of water and other materials in and out by osmosis, of course, and it also recycles and decomposes, breaks down proteins, fats, carbohydrates, and uses that as a powerhouse to make energy and to give fuel for your cells to be active and to motor and to perform the deeds that it needs to do. Then we have plastids. The plastids are small disc shapes. They are ovoid or irregular shaped organelles found only in plant cells. And you get three types. You get the chloroplasts, you get chromosomes, chromoplasts, and leucoplasts. Now, the chloroplasts are the green parts found in plants and algae, where the chromoplasts contains the red, orange, yellow, which are there for ripening fruits, flowers, etc. And the leucoplasts are the white plastids found in roots of a plant. Now, the chloroplasts, as I said, is usually found in plant cells, contains the green chlorophyll, and remember the green chlorophyll helps us with photosynthesis, where we use that for the plant to make food and from sunlight. So the chloroplast is your green, which helps with the photosynthesis. Then inside the cell, you get the Golgi bodies that are protein packaging plants. They move material within the cell, they move materials out of the cell. You will see that this is where proteins needs to be transported from where, or they take the proteins that have been made and transport it from where they are synthesized to where they are required. So they take it to the necessary organs or the part of the cell that needs it to function and to make certain functions work and where it is required. It also helps with sorting. It is a sorting cabinet in the body that shows that this must go there, 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 and make sure that everything is on the right place inside of the body. Then your lysosome, as I said, it's a digestive plant for proteins, fats, and carbohydrates. It removes waste. It takes it out. Everything that is unnecessary, that isn't needed in the body, that is actually just waste and just overall extra. It helps you to also break down the cells. Cells break down if lysosome explodes. So if there's too much and then it just breaks down and then your cell's old and expired. The lysosome is formed by the Golgi body, which is very interesting, and it contains powerful digestive enzymes, really to get rid of all the unnecessary waste that is there. It also helps with digestion, as I said, and so this is like your wastage box, if I can say that. Then your vacuole membrane bound sacs for storage, digestion, and waste removal. Your vacuole is there to remove certain things, make sure that it's not there as well, with the Golgi body as well. It contains water solutions. It helps maintain the shape, which is an extremely important part of the vacuole, and it's found only inside of the plant cells, only inside of the plants. So if you're going to look at the vacuole on the side here, this is a very nice picture just to go through everything that we have discussed so far. You'll see that the vacuole also fits in quite a big part of the cell. It takes up a lot of space. The cell wall is on the outside of the plant, which protects it, as we said. Then the cell membrane is the little blue line on the side, which helps with osmosis, letting things in and out. Also with your, it's also flexible, which I didn't mention before, which allows for it to grow and to become bigger, etc. Then the lysosomes, which we just spoke of, is in there, very small. The cytoplasm, the gray area, which is there for the liquid, where everything takes place inside the body. The chloroplast, which is there for the greenness, photosynthesis. The Golgi complex, digestion. The endoplasmic reticulum, do you see it's also the endoplasmic reticulum forms a covering over the nucleus. It's around the nucleus. As we said, in grade 12, you're going to do protein synthesis and how the nucleus and the endoplasmic will go from your DNA to your RNA. So that is why they are so close together. Then your mitochondria, which is your powerhouse, your tool, etc. Food, it gives you the food for being active. Your nucleus on the side, and then your nucleolus on the inside. So that is a basic picture and cell structure. Which I didn't say is your vacuole also takes in and releases water by osmosis. So it also has that type of function. The vacuole maintains the shape of the cell. It's big enough to help maintain that it doesn't shrink and become bigger, etc. It also exerts pressure outwards, and that pressure is called the turgor pressure, which makes sure that there's enough water inside that it keeps it at a certain place. If there's too little, the cell function will not be controlled and will not be normal as it should be. So the vacuole also plays quite a big role, just like the nucleus. Now, the cilia and the flagella is the functions in moving cells, in moving fluids, or in small particles across the cell surface. So it helps everything to move inside the cell and to function. The cilia are shorter and more numerous on cells than the flagella, and the flagella are longer and fewer on cells. They are the tail at the back, basically, which helps the cell, and there around you can see the cilia on the cell. So there was everything inside the cell and what it contains and where it, the functions are, the structure of it, and just basically an idea of what the cell looks like and what it is all about. Hello grade 10s, we are back. So we went through all the functions and the structure and how it looks and all the parts of it. So we're going to continue now with part 2, where I got a few questions for you out of previous question papers, how they ask it, just so you can see and have an idea of what the examiner will have and expect from you when answering the certain questions, etc. So you will firstly see that I have a part 2, and I'm firstly going to quickly discuss the differences between a plant and an animal cell. There's quite a few differences, and sometimes they can also ask you to tabulate a few differences, where you have to tell them, yes, this is this and this is this, and this is what makes the two different. There are a few similarities, but these are the differences that I want us to go through. So firstly, that animal cells do not contain plastids, where plant cells, on the other side, almost all plant cells contain plastids such as chloroplasts, chromoplasts, and leucoplasts. Remember, that is the red, green, and yellow and white part of the plant, which makes all the colors in the plant. Then an animal cell does not have any cell wall, where a plant cell has a rigid cellular cell wall in addition to the cell membrane. Remember, we said the cell wall makes it strong and protects it from the outer side, etc. Then it contains centrioles, the animal cell, where the plant cell do not contain any centrioles. In the animal cell, it has a large, or it is large, with a regular shape, usually round, etc., where plant cells are very small and they are irregular shape. There's not a fixed shape for a plant cell. Then animal cells do not have plasmodesmata or pits, where plant cells do. Then only few animal cells, if any, have a vacuole. I would actually say none, but I think there are a few, where plant cells, as I showed you earlier, have large central vacuoles filled with cell sap in mature cells. Then the animal cell has a nucleus and is generally found at the center of cytoplasm, and in plant cells the nucleus is found near the edge of the cell. So it's on the inside at the one and on the edge of the cell in the other. Then in the animal cell, there's no intercellular spaces found between the cells, where in plants there's large intercellular air spaces found between the cells. Now, the animal cell also has cell membrane such as microvilli, where the plant cells has no microvilli. The animal cells has a centrosome present in most animal cells, and where the plant cell has no centrosome present, it's absent. And then animal cells have lysosomes clearly visible, where plant cells, it is not clearly visible. So that is just a few differences between the animal and the plant cell. Some of them are very important, and I think a lot of cases they do ask you, please tabulate a few differences between the animal and the plant cell. Then I made gathered a few questions for you on previous question papers, and I want us to discuss it at the moment and answer it and go through it and see what it was expected from us in a test. So this was a section out of section B, question 2. It said, study the diagrams below representing two cells with their organelles. So they gave us cell X and cell Y, and they told us that we have to answer the few questions. You would see usually these type of questions are short. It's section B questions, so it's shorter type questions, but not as easy as multiple choice, etc. So firstly, they asked us which cell, X or Y, represents a plant cell. So you had to go and see all the different characteristics, etc., in both cells to decide whether it is cell Y or cell X. So if you're going to agree with me that our plant cell would be cell Y. And then they ask us, give two visible reasons for your answer. Now you can agree with me that firstly, you can see the large vacuole which is present. And remember, only a plant cell has a vacuole. So we can say because it has a vacuole, you can also include large because it is quite large. You can say it takes up quite some space inside the plant cell. Then you can also agree it has a cell wall on the outside, presence of a cell wall. And then if you're going to look clearly, it has chloroplasts. So you can say contains chloroplasts. Please, it was only necessary for two, but any one of those you could have said and it would have been marked. Usually in a test, they will only mark the first two because that is what they asked for. So even if you're going to write 3 or 4 or 5 things for them, they are only going to mark 2. Then they asked that we have to label A and B. If you're going to look at A, you would agree with me that it's the mitochondria. And if you're going to look at B, you can agree that it's the endoplasmic reticulum. So do you see inside there's our nucleus with a chromatin network, our nucleus, and around is our endoplasmic reticulum. Good. So then you have to label. So please make sure that you also know how to label a plant and an animal cell, because that can be asked of you. Then the next question stated, structure C plays an important role in cell Y. So the C, and if you can agree with me, it is our large vacuole. Now they said, list 3 functions of this structure. Now we went through the 3 functions, but I'm going to also let us write some of them down. So you can also write some of the vacuoles functions down to make sure that we know what it is. So firstly, you would agree that it stores water. It also stores organic and inorganic substances. You can also agree that I said it releases a pressure called the turgor pressure. So it ensures that the turgor pressure is there to support the cell, especially in young plant cells. Then it also helps with a high concentration of solutes in the vacuole. Increases by the uptake of osmosis. So it helps us with all of those different types of things. So please make sure that you know that. Again, they ask for 3 functions, so make sure that they're going to mark the first 3. So again, don't waste time on giving more than 3 functions. Give the 3 that is needed. Then the last question was, name the organic substance that part D in cell Y mainly consists of. So what does the cell wall mainly consist of? And what it consists of is cellulose. So that is what it's made up of. So make sure you know everything about the cell. That was question 2 for 9 marks. So there was an added question 3 on this question. And I just want to go because this is a bit more practical, as I said when we did osmosis. So the picture explains the diagram below illustrates a process through a differentially permeable membrane, letting things or allowing things to go and move in and out from the organism. So the first one said, a cell containing a sugar solution placed in a beaker with water. So that was figure 1. Our sugar solution is that on the inside, then the membrane and the water particles. Then the second one is a cell containing water placed in a beaker with a sugar solution. So you will see again that the sugar solution is on the outside of the membrane. Now the first question here, they just want you to understand and know from you, what do you know and what do you see as differentially permeable membrane? What is a differentially permeable membrane? So if we're going to look at that, it is a membrane allowing only certain substances to move through and not others. So it's only going to allow certain things to move in and out. So it makes the distinguish between what is needed and what is important and not. So please make sure that they want to know, do you understand what does it mean when something is differentially permeable? Then the next one said, describe what happens in figure 2 and explain your answer by indicating in which direction the water is moving and why. So they want to know what's happening in this figure. They also want to know, is the water moving out the membrane or into the membrane, and why is it moving in that specific direction? So if we're going to look at it, you will agree that the cell is shrinking. And because the cell is shrinking is because of the water moving out. So you can agree that the water is moving out to the sugar solution. So firstly, we can say that the cell is shrinking, and that is because the water is moving out of the cell, of the membrane. They also call that an Exo Osmosis because it's moving to the outside, the exterior. And now you would agree that the water potential, when it's moving to the outside, the water potential on the outside of the cell will be less than the water potential as on the inside. So you can say the water potential in the cell is higher because it pushed the water out, then the water potential on the outside of the cell. So you can also agree if the water moves back in, the outside potential will be higher than the inside. So it works vice versa. If it's higher on the inside, it pushes it out. If it's higher on the outside, it pushes it to the inside. And then the last question says here, which physiological process is represented here? And it's osmosis. So osmosis is taking place, moving things into and out of your cell membrane. So please make sure of that you make sure that I understand, yes, things are going out, things are coming into the cell, etc. Also make sure that you understand what does it mean for something to be permeable. Differentially also means, remember, it decides. It decides, it's not just permeable for everything, it decides what can be allowed in, what is needed, what is important. So please make sure that you understand that. That was our first question, question 2. Then you will see that we are going to move to another question, question 1.4. It says that a grade 10 learner submitted the following drawings of animal cells and plant cells that he viewed under a light microscope. Remember when I said it's extremely small and microscopic, so it's not visible by the naked eye. You have to look at the microscope. So these were the two that he investigated and that he saw. And your first question again is, they want to know which one of the following, A or B, represents a plant and an animal cell. Now, if you're going to look at it, you will agree that our A is going to be our plant. You will see it's much closer to one another, and this is our plant cell. And then number B is our animal cell. So you can agree animal and plant cell. Then they ask for state 2 visible differences between cells A and B. They want you to state what is the difference between these two. But please, there are a lot of differences, but they only want the ones that you can see on the picture at the moment, which are visible. Don't go and say differences that you just know by studying it. You have to be able to, at the moment, see the differences and then explain, this is the example or this. So if we're going to look at the two differences, most of the time it's easier to just quickly tabulate it and say what's the differences. So if we're going to say our plant cell and our animal cell, and you can agree number 1 is the plant cell has a cell wall, where you can see that around here there is no cell wall. So one thing is a cell wall for the plant cell, and in the animal cell, no cell wall. Then number 2, you can also agree, do you see there was a regular shape in these cells? In this one, there were no shape, irregular. So there were different kinds of shapes. So please, again, it must be visible inside the picture. You can't at the moment say the one has a vacuole, the one not. It must be able to be seen on the drawing at the moment. Then number 1.4.3 asks for state 2 visible similarities between cell A and cell B. Now when you do that, remember similarities means the same, what they have the same of. So it's not necessary for you then to do a table where we differentiate between the two. So firstly we can say that there is a nucleus in both of them. So there's a nucleus found in both cells. You can also agree that both of them have a cell membrane. A cell membrane is found in both cells. And then cytoplasm is found in both cells. You can agree that all around here and all around there will be cytoplasm visible. Which is very important. Then they said mitochondria and chloroplasts were not visible under the microscope. Explain why this is so. Because remember, this is what you saw under a light microscope. Okay? He saw that underneath the light microscope. They wanted to know, why is it not possible for us to see the mitochondria and chloroplasts in these two? So you can agree that because it was underneath the light microscope cannot see structures that are so small. So if it was another microscope, maybe like an electron microscope, then it would be possible. But because it was under a light microscope, it's too small to be seen. So it's not visible, 1.4.4, sorry, not visible because too small to be seen under a light microscope. So please remember that, it's still too small. Even though the cells are also small itself, the mitochondria and chloroplasts are even smaller. And then the last question in this section, you will see that state 2 functions of the part labeled C. So again, we have to make sure that what is the part labeled C? Now remember, we said that was one of the similarities in our question, which was our nucleus. Now our nucleus, you can first maybe just say yes, it is the nucleus again if you want, and then you can just state 2 similarities. Now remember that the nucleus controls the metabolic activities of the cell. So you could say control metabolic activities of the cell, as well as it transmits genetic characteristics from cell to cell. Remember when we discussed this, we said that you can find your heritage from parent to offspring in cell to cell. So it transmits genetic characteristics from cell to cell. So it gives you the genes from your parent to offspring to see the direct link from your parents and your genes that's maybe been inherited, your similar dimples, your eyebrows, your earlobes, etc. So that you can also be seen for that. Parentage, to see, is your father your father? Do you have the same DNA as your mom? So the nucleus also carries that genes and that gene pool that is extremely important in a human being. That was question 1.4. That was still part of the first question, which is the the shorter questions. So you will see that it was shorter questions, and in this one as well it was 1 mark, 2 mark questions, and for each one there was a specific question that was asked. So if we continue, we will see that we're going to do a bit of more in-depth questions. We are going to do longer questions, essay questions, which is not going to be 1 or 2 marks, which is going to ask you to explain it in much more detail than just the basics that you need to know. So please make sure that in the next session we will have a bit of deeper and more insight questions that must be answered. I hope you had a lovely class. Thank you.